Hello dear learners, in our today's class and from our in this video and from this video we are going to see and solve some problems in genetics, that is particularly the numerical problems which are popularly asked in the form of questions in genetics. So first of all, in today's this video I will first of all talk about the 3-point crosses, where we popularly call them as tri-hybrid crosses. So in the tri-hybrid cross, basically the numericals arise out of the concept of linkage, and we have to discuss that in our today's class. So how can we solve these questions? So let me take the hypothetical example. Suppose this is your chromosome, a particular chromosome or a segment of chromosome, and herein you have three different genes. Let's say one is of blue color here. Okay, wait. Let's say one is of blue color here, then another one is of red color here, and then there is pink colored. These are the three different genes which are present on a single chromosome. Let us name these genes as, supposedly, let's say gene A, sorry, let's name them as gene A, B and C. Now what happens is that we have learned the Mendel laws, and according to Mendel laws we know that there is the law which is the law of segregation, and then there is another law, that is law of independent assortment, right? So according to the law of independent assortment, what should happen while this is particularly being crossed? What I'm saying is, while this particular, let's say the segment of chromosome is crossing over with another segment of chromosome, wherein we have, suppose, let's say different form of alleles. Instead of A, B, C, let's say we have capital A, capital B and capital C. So now what happens is basically that uh, in the crossing over generally between a single allele and a single gene, ideally the crossing over should take place, and here capital C should come and here small c should go. That is, the alleles must be exchanged. That is what we call as crossing over. But in the concept of linkage, what happens is that if the distance between the two of the genes is very less, then they tend to segregate together, means they will not recombine and they will not show the crossing over. In that condition, the recombination is less. So I can write here the recombination here is less, and the parental type even in the offsprings is higher. So this will be happening when there is more linkage. And when the two genes are less linked, means the distance between the two genes is higher. That time the linkage will be less, and that time the recombination chances would be higher. So that is what we call as a recombination in the, suppose, let's say the two different genotypes of the parents, or the two different parental genotypes. Now when we are considering the tri-hybrid crosses, there might be chances that if the two genes are linked in a particular parent, then in the progeny also, when these two are crossing in their progeny, the two of the genes should be linked. So in the case of linkage, the recombination would not occur. Let's understand with this with the help of a simple example. So supposedly I am saying that here I have, suppose these are my two chromosomes, and here I have the genes like, suppose, A, B and C, and here I have their wild types. Okay, now what happens is, suppose I say that the B and C linked, even their wild types, and they are closely linked. Means what I'm trying to show is this B and C are very close to each other. That time they have the linkage in between them. So when they will be showing the crossing over, only this part, if I were to highlight this, only this part would show the crossing over, right? So what would be the resultant progeny be like? So here, if I say the resultant progeny would be like, here I will get plus, here B and C, and here I would get A, and here plus and plus, the wild forms. So this kind of process wherein only a single gene is changing or recombining. Now this is referred to as, this process is referred to as single crossover. Single crossover. In single crossover, only one gene is changing. Now, if you try to look yourself, if you try to imagine it, it can also be the way that, suppose, just give me a minute. I'm just copy pasting the two parents again, okay, and this I'm again bringing here only. Now let's say this B is close to A and the C is away from them. Now what can happen? What are the chances? The chances are that the C will be showing the recombination, right? So in that case, what we are going to get as our progenies, we would get a condition or the progenies wherein A and B are linked, but here we'll get plus. Similarly here plus and plus, but here we'll get C. So this condition is also single crossover. Getting it? So this is also single crossover only, and this is also single crossover. But for the name and for the simplicity, if the two genes which are located firstly, if they are showing the crossovers, then the progenies are referred to as single crossover 1. So we call them as SCO1. And if it is about the last pair of genes who are recombining, that time we call it as single crossover 2. Then there are certain other chances also. The other chances may be that suppose we are having this parent and here, wait, let me copy paste this again. Here what is happening now, the difference is that here in the parent also all the genes are equidistant from each other. In that case, what can happen is that this would also have the chances to show the crossing over, and this would also have the chances to show the crossing over. So in this scenario, we will get a condition wherein the two genes are showing the crossing over together. Okay, so I'm naming it here. This is SCO1. In this case, we will get the recombinants wherein we have the two crossing overs at the same time. So it would be like this. This is the case where we call it that there has been double crossing over which has taken place. So these are the double crossovers. The progenies which have the double crossing over are called as double crossover or simply DCO. So these are very important and to be known first, because if we do not know the concept of single crossover 1, single crossover 2, double crossover, we would not be able to solve the questions. Now let's come on to the questions and how do we find out and what are the formulas. All of them I will be telling you once I start solving the question. Let's look over this question. The question says in Drosophila, the recessive mutant allele, that is cinnabar. This is one of the alleles, and the CN is the gene. It results in bright red eyes, and curve C results in the curved wings, and plexus PX has extra wing veins. So there are three genes which are being considered here. One of them is CN, one of them is C, and one of them is PX. Now data is provided, and in typical questions like these in genetics, then when the numericals come on linkage, so this kind of data would be provided. Now in this data, what you need to remember is that when if such kind of genotypes are observed or are given to you, you have to just ignore these because all of them are same. What you are concerned and what you should be concerned is only about these genotypes. So what we need to know is that we have to just focus on these genotypes and their respective numbers. So let's start solving them one by one. Okay, I'm zooming it so that it would be more simple. See, now first step one is that, okay, it is much more darker. Step 1 is identify the parents. Now, who are the parents? See, the parents are those which have the maximum number. So, I am trying to represent them with the green pen. Here, 296 and 329. These are the parents whose number is maximum. So, we will directly assign them that this is also and can be a parent, and this also can be a parent. So, I have two parents here, that is CN, C, and PX, and then it's wild form CN plus. Okay, let me write it here only. Wait. Cn plus C plus Px plus. Their numbers are 329, 296. Now, one more key feature you need to know is you just look over all the genotypes. See here CN is also covered and its wild form. C its wild form. PX its wild form means all the six genes are present here. No. So when you talk about the parents, the six genes must be covered. So you have to choose only those two maximum numbers which consist of all the six genes. See, this is gene type 1. This is gene type 2. This is gene type 3. This is gene type 4. It is different from this. It is the wild form. Na? Then this is gene type 5 and this is gene type 6. The first step was the identification of parents. The second step would be identifying the double crossovers. Now double crossovers are very easy to identify. I'll mark them in pink. All those progeny which are least in number will be the double crossovers because the chances of recombination at two genes is very less. So those will be the double crossovers and they would be very less in number. So this 10 and 15 would definitely be the double crossovers and they will be represented like CN, C plus, PX, C. Don't get confused, this means C plus, the wild form of C. And then I have CN plus, C and PX plus. Now their numbers are 10 and 15. Next step is, and the first thing is that we have to deduce the order of genes. So how to find the order of genes? See, the step is very simple. You have to focus only on the double crossovers. Okay. Now you take one gene common in them, which if you try to interchange, you get this parental genotype. Again, I am repeating. In the genotype of the two double crossovers, take one gene, which if you interchange, you get the exact parental genotype. So if I interchange this C plus and C, so I can get the exact parental genotype. So that gene whose interchanging is resulting into the exact parental genotype would be always in the center. Now in this question it was simple that C was the one, but in another questions it would be different. Might be possible that PX was there. So if you're interchanging PX and if you're getting the parental genotype, it means that PX is actually in the middle. But here because C is only the answer, so this is the correct genotype. We can write it as CN CPX or any form of this. C should be in the between. So this is the first answer, that is we have to find the gene order. So I can find the gene order clearly. The gene order will be CN CPX. I'll mark it like this. That is, it is on the chromosome. Next is that we have to determine the map distance. Now how to determine map distance? Map distance simply means that we have to find the distance between the CN and C, and then we have to find the distance between C and PX. Now this is another important thing which you should know how to solve. So how can we find the map distance? So let's find the map distance between CN and C. It's part 2 na, so it's part 2's first subpart, CN and C. We have to find the map distance between the two. So because now we are finding the map distance between CN and C means the first gene involved. So the formula is the map distance is nothing but it is equal to number of single crossovers 1 plus double crossovers upon total into 100. That is its formula. Now you have to identify that who all are single crossovers in the above data. See, what you need to do is you got your gene order, it was CN, C and PX. Now in this gene order, write your parents. Your parents were these two, so they have already been written. The work is very easy for you right now because the question is simple. So it is CN plus, C plus and PX plus. Now remember the concept of single crossover. I told you in the single crossover only the first gene is interchanged. So if you are interchanging these two genes, what is the resultant progeny? It is CN plus CPX. Similarly, it is CN C plus PX plus. So these type of progenies, if they are mentioned in the table, add up their numbers. So Cn plus C and Px, is it there? Cn plus C and Px, yes, this is the one. And then we have another one, Cn C plus Px plus. So yes, this is the one. So 119 you have to add, and then you have to add 86. So add the two of them. So let me add. The answer comes out to be 205. I have just added single crossover once. Let's remove this so that you are not confused. We are only right now talking about this. So I've added the two of them and I'm getting 205. Now I'll put this in my formula. So the map distance will be nothing but it will be equal to 205 plus double crossovers. How many were my double crossovers? They were 10 and 15, so in total 25. So I'll write here 25, then I'll write total. Total is my, here it is given, it is 1000 multiply by 100. So if I am crossing it, I get 5 is 0, 2, then 3, and then here 2, 230 upon 10. So it's approximately 23 centimorgan, or we can write 23 map units. Any answer you write, the units are correct. So this is the distance between the CN and C. Now if I talk about, we have to find the distance now between C and PX. So I can easily solve it from again the parental genotype. Now what I'm doing is I don't have to do this one, no. This time what I need to do is I have to interchange the two of them, these two, or directly from here also you can get the answer, because now the others have been already occupied. So now here your single crossovers 2 would be the 63 and the 82. The two of them would definitely be your these. So now we should consider the two of them, 63 plus 82. So add them up, it's 145. So solving here, single crossover 2 numbers, that is 145, plus double crossovers, that is 25 upon 1000, the total number of progenies, and they multiply by 100. So it is 145 plus 25 upon 100, sorry, upon 1000, means these are the total offsprings. It would be either provided in the data or else you have to solve it yourself. Multiply by 100. So let's solve this. 4 5 6 7 and 1, 170, and this. So the answer would be 17 centimorgans, or 17, this one, map units. So this is your answer. Now what you need to do is very simple. Now you have to create the linkage map. For that you will draw a straight line, then you can even use three different colors. One is for C, wait, one is for CN, then one is for C, and then one is for PX. See, the distance between CN and C is more, so I am hypothetically creating them at a farther position, and it is 23 centimorgan. So this much is, this unit is 23 map units, and this much is 17 map units. You can even total this, it is 40 map units. So this way you can create the linkage map. Now let's see what are the other questions they are asking. The other question is you have to calculate the coefficient of coincidence and interference. Now this is important. See, what is the formula of coefficient of coincidence? Coefficient of coincidence is observed double crossovers, means all those which are given in your data, upon expected double crossovers. Now expected double crossovers, how you need to find it is very important. You need to learn it by heart. Expected double crossovers is this one. It is the frequency of single crossover 1 multiply by frequency of single crossover 2 multiply by 1000. Now how to find frequency? So frequency is simply what? Frequency is, suppose I am talking about single crossover 1. So the number of individuals of that single crossover, how many were they? They were 205. So 205 upon total means how much times they are occurring. So it's approximately 0.20, right? And for single crossover 2, it's 145, right? Yes, total number of single crossover 2 are 145 upon total number of individuals are 1000. So it's 0.14. So what you will write here, it is equals to Observed double crossovers are nothing but these are 25, right? They are given to you 25. Yes. And then the expected double crossovers, as I right now only told you, they are 0.20 multiply by 0.14 multiply by 1000. So it's approximately 28 and upon is 25. This one is 25. So it's approximately 0.89, and this is valid because when I talk about the coefficient of coincidence, it should never be greater than 1. If it's greater than 1, then your data is somewhere inaccurate. Then interference is 1 minus coefficient of coincidence. So interference should be 1 minus 0.89. The theoretical knowledge of interference and coefficient of coincidence, I assume you know. So I'm just solving it in the form of numericals. So it is approximately 0.11. So this is the answer. So more or less these kind of questions would only be given in your exam, and you can easily solve them just by calculating the data. You just need data, and formulas should be learned to you by heart. In that condition, you can easily solve these questions.